Hello, everyone, and welcome to this special oncology webinar presentation where you, the audience, gets to pick the brains of some of the members of our global scientific applications team on what the best practices are for your cancer imaging and data analysis. My name is Sarah Burris. I'm the global leader of scientific affairs and applications here at FUJIFILM VisualSonics, and I'll be moderating today's webinar. At this time, I'd like to welcome the other members of the panel to introduce themselves, starting with Drew Heinmiller.
Hi, everybody. My name is Drew Heinmiller. I'm the Acting Director of Product and Marketing here at Fujifilm VisualSonics. Coming to you in Toronto, where we've got a little bit of snow on the ground, appropriate for the season. I've been with the company for a long time now. I started as an application scientist working with our first photoacoustic imaging platform, the Vevo Laser. And in that work, there was quite a bit of oncology applications development that went on, including looking at tumor oxygen saturation, vascularization, and using various photoacoustic contrast agents to do some molecular imaging. So yeah, thanks for having me.
Thanks, Drew. And now Philippe.
Thanks, Sarah. It's a real pleasure to be here. I'm Philippe Trochet, Scientific Application at Fujifilm VisualSonics. I must say thanks to the researchers because I gained a lot of experience on many different application areas, and more especially in oncology, working at VisualSonics for the last 17 years. So it will be a real pleasure to answer your questions after Sarah's presentation.
Thanks, Philippe. And Anna.
Hello, good morning. I'm from the West Coast in the US, coming to you from Portland, Oregon. I've been with VisualSonics for 2 years, and I did my dissertation developing contrast agents for photoacoustics, specifically in the realm of ovarian cancer.
Great. Thanks, Anna. Well, just a few quick notes about the webinar today. A recording will be made available to those of you that registered. And as a reminder, all the lines are muted for the duration of the webinar. So if you have a question for the experts, please enter it in the Q&A button, and we will answer those in just a few moments. So as we wait for some of the initial questions to come in, I wanted to take just a couple minutes and tell you about some of the latest innovations at Fujifilm VisualSonics and how we can help potentially expand your research. So I'm sure most of you are familiar with the VisualSonics product portfolio seen here, but we've been the industry leader in preclinical ultra-high frequency ultrasound and photoacoustics for more than 25 years. And during that time, we've released several iterations of our preclinical ultrasound system, leading up to our flagship system called the Vevo F2 that we released just a few years ago. Now, this system has several key features that distinguish it from our previous systems, in that the hardware on the Vevo F2 allows you to run both high and low frequency transducers as high as 71 megahertz, as low as 1 megahertz, which ultimately enables you to image something like a heart in a 70 kilogram pig, as well as a heart in something as small as a zebrafish. Now, similarly, this system can also accommodate phased array transducers, which allow you to get a better acoustic window in larger animals and some disease models. This system also has faster frame rates, which allow us to visualize dynamic processes. It also has enhanced Doppler sensitivity for better blood flow visualization. The F2 also has modes that aren't necessarily available on previous systems, such as 4D color Doppler and shear wave elastography that I'll discuss in just one moment. And finally, the system also has something called VADA mode, or Vevo Advanced Data Acquisition mode, which if you're in acoustic engineering, this mode allows you to access the pre-beamform individual channel data through a very easy to use graphical interface. Now, regardless of the system that you have, you've been able to acquire a variety of images and data sets in multiple organ systems and animals. But of course, this is an oncology-focused webinar, so I wanted to provide you with just a little bit of information on the different ways the Vevo F2 can help you expand your cancer research, starting with how you induce your tumor models. Now, our system comes with a small animal rail system, which has several benefits in terms of monitoring the animal's health and keeping the animal warm. But in terms of actually inducing your tumor models, it has an optional injection mount system seen here on the left-hand side that really enables precise, repeatable injections of tumor cells in your area or organ of interest. Now, the benefit of this is that you don't have to use invasive surgical procedures. You can actually visualize the needle as it enters into the body. And there isn't really a need for post-surgical analgesics, and the recovery time is substantially shorter. Now, in terms of throughput, this is significantly faster than traditional surgical procedures. And as shown in this particular paper, you can actually get more consistent tumor placement and more consistent tumor sizing when using this technique as opposed to traditional surgery, thus potentially reducing the number of animals that you need for each study. Now, once you've injected your tumor cells, you're going to want to start to watch them grow as soon as possible. Our system allows you to visualize these tumors when they're as small as 30 microns. And of course, if these tumors can be detected far before they're palpable, then the results of your experiment may have more translational relevance depending upon the model. Next, you're going to want to get accurate tumor sizing as the disease either progresses or potentially regresses if you're testing a specific treatment. Now, you have the ability to do what we call in science, the quick and dirty measurement of 2D imaging. And we have some fast analysis tools that either allow you to look at the total area of the tumor or get linear-based measurements. Now, of course, the formulas to determine area off of this are really based on a very spherical shaped tumor. And we know that tumors don't necessarily always have that homogeneous form. So the real benefit of our system in terms of tumor volumetrics is that you can acquire multiple 2D images using this 3D motor shown here over the length of the tumor. And this, of course, gives true volumetrics of that tumor. Now, depending upon the modes that you have on your customized system, you can also assess hemodynamics of the tumors. This is accomplished through things like color Doppler and power Doppler. Now, these modes give us valuable information about the velocity and direction of blood flow in 2D. Now, in 2D, these are kind of purely qualitative measurements, but when you pair this with either pulse wave Doppler to get things like velocity of feeder vessels into the tumor, or acquire these color Doppler or power Doppler in 3D, they become quantitative. In the sense that you now can get things like percent vascularity of the entire tumor. Oops. Now, Doppler imaging modes are great for visualizing large vessels, but if you want to assess things like perfusion or see small vessels such as the capillaries, you're going to need to use something called nonlinear contrast mode. Now, this mode combines something called Vevo micromarker, and Vevo micromarker is a contrast enhancement agent that's roughly 2 to 3 microns in size that, when used in its non-conjugated form, can be injected through the tail vein, and it circulates throughout the body. And it really can be used to help visualize blood flow and perfusion of a tumor or a specific organ of interest. We also have advanced analysis software that can be used with this Vevo micromarker, here Vevo CQ. And this really allows us to look at multiple parameters, things like peak enhancements, area under the curves, rise time, time to peak, and so on. Now, I realize this isn't a cardiac webinar, but I'd really be remiss if I didn't mention the fact that you have the ability to assess cardiac function with the Vevo F2. In terms of cancer treatment, we know that maintaining cardiac function is incredibly important. If a physician has put someone on chemotherapy and it's damaged the heart, there's no recovering from that. So monitoring early changes in cardiovascular function is essential to know that your drug is, of course, working on the tumor, but also not damaging other parts of the body in the process. So Vevo Strain 2.0 is a great piece of software that actually acts as an early detection system for looking at cardiac dysfunction far before you have any decrease in, say, parameters like ejection fraction. Now, one of our newest imaging modes available on the Vevo F2 is shear wave elastography. And this is a non-invasive imaging technique used to look at tissue stiffness during disease development. Now, it does this by essentially introducing a low frequency vibration or a shear wave into the tissue that then gets detected with ultrasound. Now, shear wave speed is essentially used to determine the stiffness of the tissue, in that the faster the wave propagates through the tissue, the stiffer that tissue is. And of course, we can use this to analyze things like tumor stiffness, but it's also a great technique for looking at things like liver fibrosis or potentially changes that occur in the kidney, as well as changes that occur in the musculoskeletal system. Now, very recently, I want to say almost 2 weeks ago, there is a great publication that came out utilizing shear wave elastography and a MASH model of liver fibrosis. And what they found is that shear wave elastography measurements on the Vevo F2 correlated very nicely with the gold standard MRE detection of fibrosis, as well as histological assessments of fibrosis. Now, you may have seen in one of our previous webinars over the last few months that we recently introduced a brand new photoacoustics platform called the Laser X20. Now, this system utilizes the same flexible light delivery as our previous system, the Laser X, in that you can choose which transducer you'd like to pair with the appropriate fiber to do photoacoustic imaging. And for those of you that aren't as familiar with photoacoustic imaging, this is essentially a light in, sound out process. It basically delivers nanosecond pulse laser light from these fiber cables. That laser light gets absorbed by chromophores. This causes thermoelastic expansion in the tissue. That thermoelastic expansion generates a sound wave, which we then detect with our ultrasound transducer. This, of course, giving us the benefit of getting molecular information on top of all that functional and anatomical data that you're collecting with the ultrasound. Now, this can be accomplished just with endogenous chromophores. Things like melanin and hemoglobin are natural absorbers of photoacoustic light, but you can also inject things like near-infrared dyes and photoacoustic reagents like nanoparticles. And based on what their absorption profiles are, we may know what that is. For the example of hemoglobin, we can do what's called unmixing for these signals to then display highly oxygenated areas of the tumor, shown here in red, versus other areas that don't have a lot of oxygen concentration, displayed in blue, as well as where a nanoparticle has distributed throughout the tumor, depicted here in yellow. Now, as I mentioned, the predecessor to the X20 was something called the Laser X. That is a slightly smaller platform than the Laser X, but the added benefits that you get from this system is that it has improved sensitivity. So here we're just looking at a subcutaneous tumor image with the Laser X versus the X20, and the green signal that we see is actually circulating ICG, and we can see far greater signal in the tumor image with the X20 than that image with the Laser X. Now, in addition to having higher energy and greater sensitivity, this Laser X20 also has a new expanded wavelength range for detection. Now, our previous system went from 680 to 970 and then had a gap in its acquisition, and then you could image again from 1200 to 2000. The X20 actually allows us to image slightly further down the spectrum, so starting at 660 nanometers all the way through 1320. And of course, the added benefit of this is that we can potentially start to see other compounds that reside within this range, such as collagen, and also do things like lipid detection. Now, everything I've mentioned up until this point are things that you can do solely with the Vevo F2, but in introducing photoacoustics, you get that added benefit of looking at things like oxygen saturation throughout the tumor, as well as looking at additional molecular markers that you would potentially inject throughout the course of disease progression or treatment. And I just want to say that these systems are not just limited to one type of cancer, like subcutaneous tumors that I've been showing here. We have over 700 publications looking at cancers in the brain, in the kidneys, the prostate. There's some really beautiful examples of colon cancer, pancreatic cancer, liver, and even in areas like the lung. So that's pretty much all I have for you in terms of the presentation. So at this time, I would love to start turning it over to our panel to answer some of the questions that have come through in the chat. So at this time, I'll just ask the other members of the panel to please turn their cameras on. Thank you. And we'll go ahead and begin. So just as a reminder, please enter all of your questions in the chat and we'll get to them as soon as possible. So I'm going to start out with the first question that has rolled in. What types of tumors can be detected in rodents using ultrasound and or photoacoustic imaging?
Yeah, I'll jump in here. Basically, um, any tumor type that you can induce in a small animal model, so mouse, rat, and as Sarah mentioned, larger animals as well. Um, I would say, some people say, oh, well, ultrasound, doesn't go through air. So what about lungs? Well, there's actually been a group who's actually even done lung tumor imaging where they had a model that was induced on the surface of the lung and you were able to see the tumor growing in the lung. So even lung tumors are some possibilities here. So all sorts of different tumors, I would say. Some of the advantages of the ultra high frequency ultrasound that we provide are really detecting those very, very small tumors, particularly orthotopic tumors. So things like pancreatic tumors, liver, kidney, ovarian tumors, prostate, bladder, all of these tumor models that orthotopic tumor models, especially at that very, very small stage, pre-palpable, that's all, the advantages of ultra high frequency are really, really obvious for those kinds of models.
I want to jump in here and say that I saw a really great example of using an interesting cancer model. It was actually done with 3D spheroids. So it wasn't even just using cancer the way we would normally think of it, using subcutaneous or orthotopic, but it was actually doing 3D spheroids grown in a petri dish. So that was another cool application that I saw recently.
And in addition, I should say that sometimes you may think that it's very challenging detecting brain tumors, but in reality, it's something feasible using our unique imaging platform combining high-frequency ultrasounds and photoacoustic imaging. As an example, you can differentiate primary brain tumor from secondary brain tumor. The primary brain tumor is the melanoma brain tumor. The secondary is the melanoma brain tumor, and by detecting the melanoma content, the melanin content into the tumor, you can detect it in photoacoustic imaging mode. And for the glioblastoma tumor, this is something you can detect as well in photoacoustic using a specific targeted organic contrast agents targeting the alpha beta 3 integrins. So this is also part of the studies you can do for detecting brain tumors.
Great, thanks. Thanks, everyone. Can you share specific examples of how photoacoustic imaging has improved the monitoring of the tumor response to therapies, such as anti-angiogenic treatments?
There's many examples of this, I would say, many of which have been published. So you can check out our publications library on our website to see some of that. One example just off the top of my head is an example from Dr. Malidi, sorry. And she was doing photodynamic therapy and looking at the effect of photodynamic therapy. She was specifically looking at the relationship between the oxygen saturation status, so really the hypoxia status, which you measure using photoacoustics, and how that influenced treatment. And there was a couple of interesting things that she found. One was that it wasn't just the overall oxygen saturation status of the tumor she was looking at. So again, with ultra high frequency, you get very, very high resolution. So you can even look at small tumors, even, less than 2 millimeter tumors, and look at sub-regions within those tumors. And that's the work that she had done, where she noticed that, in highly oxygenated areas of a tumor, those areas responded well to the photodynamic therapy treatment. The more hypoxic areas of the tumor didn't respond as well. And she could actually generate maps based on the photoacoustic signal, looking at oxygen saturation with photoacoustics. She could actually create 3D maps within the tumor, pointing out and predictive maps pointing out which areas of the tumor would be likely to respond to therapy versus those that were less likely to respond to therapy. Purely based, this is no external contrast agents. This is just purely based on the oxygen saturation status of the blood measured using photoacoustics.
Yeah, and I'll just add to that, Drew. We actually had a customer, Ana Rosa Arcangeli. Her group just recently presented some work at WIMIC, the World Molecular Imaging Congress, doing something similar with photoacoustics, determining how you can show kind of an area that would respond to a tumor versus a non-responding area and how that correlated to oxygen saturation. And they also showed the added benefit of having shear wave elastography in that area as well. So those areas that were dubbed kind of this non-responding area also had stiffer tissue. So it was a really nice kind of comprehensive study. So this was actually a two-part question. So the second part of that was, are there any specific methods or best practices that you would recommend for optimizing photoacoustic imaging of tumors?
Yeah, I saw this question come in, and so kind of feeding like back onto what you all were saying. One of the things I used to do when I was doing this for a PhD project was pre-characterize the tumors using vascularization, using techniques like looking at oxygenation, and then be able to go back afterwards and look at which ones responded and how they were responding because of how the treatments we were using. So one of the best practices I can share is to use all the tools you have available to you and be able to pre-characterize or go back and be able to post-characterize and look at different ways that the tumor microenvironments were interacting.
And we do have a multimodal imaging platform where you can extract many different parameters, get anatomical, functional, molecular, and mechanical informations. So that's the way how you can qualify the tumor metabolysis or part of the metabolysis.
Great, thank you. So we have a question in regards to our transducers. So does VisualSonics have a high frequency 2D ultrasound transducer? And I think I can go ahead and answer this one. Yes, we have about 7 transducers currently that pair with the Vevo F2, both linear and phased arrays. And the highest frequency transducer is a 70 megahertz transducer. So of course, that high frequency does limit the depth of penetration. So that maxes out at about 1 centimeter depth, but it does have the highest frequency and it does allow the visualization of those tumors at 30 microns. So the next question is actually related to the strain module for early detection of cardiac dysfunction. Someone's asking if this comes on the system or is this something that has to be purchased in addition to the F2? And again, I'm happy to answer this. So this is actually a software analysis tool. So it's not something that's built directly on the F2 system. It's something that pairs with our Vevo Labs software. It's a software extension. So it is something that has to be purchased in addition to the basic Vevo Labs suite. The next question.
Sorry, if I can just jump in. I think just back to the previous question, someone asking about the transducers. I should say too that so we've got linear array, 256 element linear array transducers. That all of our transducers are like that. The lower frequency curved arrays, I think, might be a little different. But one thing I wanted to add is the ability to do 3D. So we have a linear stepper motor which translates the transducer to collect basically a whole bunch of slices and then stack them together to make a volume. And that volume can extend from a millimeter, in the stack size is sort of a millimeter long, or you can do whole body imaging with that same setup. And it's really the combination of the appropriate transducer for doing something like whole body. You might use a slightly lower frequency transducer. If you're really going in organ specific and you really want to see those smallest tumors, that's when you would use the higher frequency transducers, but the 3D capabilities are available for all.
So Drew, how long do you think it takes to complete a 3D scan in B mode of, let's say, a 10 millimeter tumor?
It really depends on the mode you're trying to use, but really, literally a few seconds, a few seconds to possibly, as long as something like a minute, depending on how many data sets you're trying to collect, what that sort of slice resolution you want is, what mode you're using. So if you're using, multispectral mode in photoacoustic imaging for some of that more molecular data, it might take a little longer, but we're talking about very, very fast acquisitions. And this is really to facilitate that workflow where if you do have, multiple animals that you're scanning, every day to monitor tumor volumes, for example, and if that's all you want, 10 seconds for a scan per animal.
Yeah.
And the image quality, the 3D image quality is extremely good because you can activate triggers, the respiration triggers, as well as the ECG trigger if the tumor is close to the heart. And this way, when you scan the tumor area in a stepwise manner, when you right away after this 3D scan, you get the 3D volume construction. And you can then scroll through the different imaging plane, the sagittal one, the coronal one, the transverse one, and the quality is very good as compared as an MRI system, but much quicker to perform the 3D scan.
Yeah, and I'll just ask a subsequent question. Can the panel kind of comment on, if you're doing these longitudinal studies, kind of what I laid out in the presentation in terms of doing a B mode and then doing a 3D, color Doppler mode, can you comment a little bit on the ease of analysis in terms of once you kind of do the initial B mode analysis, as long as you haven't moved the animal and done the same 3D acquisition, the analysis is quite fast. So can you expand on that a little bit?
Oh, sorry.
Yeah.
You're talking about the. Oh, sorry. Go ahead, Blue.
Go ahead, Anna. Go ahead.
Oh, I was just clarifying. Do you mean the acquisition or the later analysis?
So kind of both in terms of once you've determined your tumor size and you've set the length at which the 3D motor is going to move, basically what I was alluding to was if you acquire that in B mode and then you move on, let's say, and you acquire 3D color Doppler, and then you acquire that same 3D spec for the tumor in oxyheme mode in photoacoustics, do you have to go in and analyze all of those images individually, or is there a way to speed up that process?
Yeah, there's a way for sure. So first of all, the animal is always positioning in the same way. We have this animal imaging station where you have the animal placed on a plate, and the plate is heated. And then the 3D motor is software controlled. So you can check the 3D scan distance, the step size you want. You can be at a very low step size if you want to have a high resolution on the 3D view, or you can increase the step size if you want to do the 3D scan quicker. And then you can apply this in the different imaging mode, in B mode, in color Doppler mode, power Doppler mode, or even in the photoacoustic mode as well, in oxyhemo mode or single of multi-wavelengths. And for the image analysis, you simply have to do the 3D volume construction on one image in B mode as an example, because this is where you have the best image resolution. And then you can copy paste the volume on the other imaging modes. And then it's very quick, and you get the volume calculation done. You get the percent vascularity done, so you know the density of the main vasculature. You can also get the microvessel density parameters as well, the oxygen saturation, so you can detect the tumor hypoxic core as well as the non-hypoxic core. and so on, or the quantity of your specific contrast agents that is bind to a specific receptor, as an example.
I would
Nice.
say too that there is a sort of an I would almost separate the acquisition stage from the analysis stage, and that's actually what we've done on our products is, we've built in all the tools. So, for example, except for B mode, every mode we have is so-called duplex mode, which means we're always collecting that high resolution B mode anatomical image, and then whatever other parameter you want to collect. So if it's perfusion, you're using ultrasound contrast or color Doppler for flow, photoacoustics for oxygen saturation and molecular imaging. And those acquisitions are essentially automated. So, you set, how much, how far do you want to acquire? I want a 1 centimeter scan along the animal in the abdomen, for example. You click go. You're collecting your B mode and let's say your Doppler, then you do your photoacoustics. And then we have our Vevo Lab software, which is our workstation software. So once you've done all the acquisition, you don't want to be tying up your scanner for analysis. So you export your data. You use our Vevo Lab software to do all sorts of analysis. And as Philippe points out, that's where you're really you've got all these data sets, but you don't have to, your measurements can be copy and pasted across the data sets. And your data sets can be analyzed in a smooth way. And you have that workflow to be able to withdraw all those measurements, like Philippe pointed out, percent oxygen saturation. You can measure, the signal coming from a contrast agent, a photoacoustic contrast agent, for example, for molecular imaging, and so on.
And I'll jump in and say that's just talking about one tumor. When we're talking about longitudinal studies, one of the nice things about both the analysis and the acquisition is that we do have presets. So it does help you kind of jump back to where you were the day before, the mouse before, wherever it is you need to be doing. So you can have more reliable both acquisition and analysis between different data sets as well.
Yeah, great. Thank you both for that. Philippe, you previously mentioned just in your description something about MRI. Can everyone comment on what the benefit of using the Vevo ultrasound is versus traditional MRI or optical imaging?
Well,
I think
MRI is a fantastic imaging modality. I must say it's great, but very complementary to high frequency ultrasound imaging, because on an MRI system, you must prepare the animal. It takes some time. And then you must also know what tumor tissues you are looking at, because you must set parameters, constant parameters in the MRI system. In ultrasound imaging, it's very quick. I mean, you place the transducer on the animal body, and then you get an image, and the image tells the truth. So you don't have to adjust and set many parameters. Eventually, you can adjust the gain depending on your eyes, and then you identify the anatomy. And because you are exercised for identifying healthy tissues from diseased tissues, right away you can detect a very tiny tumor. And we can detect tumors that are less than 1 millimeter in size, or even small lesions prior to the tumors.
Yeah, I'd echo that, Philippe, by saying, MR has great soft tissue contrast, but so does high resolution ultrasound. And I would say we exceed the resolution of MR systems, even high field MR systems, with a much more simple acquisition. You don't need an MR expert who knows how to change RF coils or set the acquisition parameters, just so, as Philippe points out, to get a certain tissue contrast. The tissue contrast is the tissue contrast. Yes, there are some settings that can be optimized, and as Anna points out, you can actually set presets for those, but it doesn't require an imaging expert. Really, most of our customers, they're oncology experts, and that's what they want to focus on, is the biology of these tumors, how effective their treatment is, how the tumor grows over time in response to treatment. And our equipment, I would say, allows that focus on the biology without having to be an expert in the equipment as well.
Great. Thank you. So we have another question that came in through the chat. So one of the participants says, I've conjugated an antibody with ICG, and I know that this antibody accumulates in tumors by recognizing an overexpressed target. When performing photoacoustic imaging at 800 nanometers, do I need to include a control mouse that has not been injected with the antibody in order to assess the background signal?
This sounds like a reviewer question. I don't know. It's so someone wants to publish, and maybe a reviewer had that. I mean, look, you're doing science. You should probably have your controls as well. So this is probably a question that the reviewer is going to ask. Great, we saw an increase in signal at a given wavelength. Uh, and that indicates your antibody. Well, where's your negative control where you have an unlabeled antibody and you don't see that corresponding signal? So I think it's not really a equipment question or imaging question. It's a science question. Um, what I would say is that again, setting up your parameters, being able to do those studies, being able to do your controls, positive control, negative control, whatever it happens to be, in a reproducible way. Again, always having that high resolution anatomy. I think that's a huge advantage here for something like optical imaging. Again, very, very sensitive, but you can't really see beneath the skin. You're just really looking at the surface of the animal. And with ultrasound, you're looking deep in tissues. You can see an anatomical context. And then on top of that, now you've got your antibody labeled with ICG. You inject that systemically, and you see it accumulate. So we have tools with our photoacoustic imaging as well, where you wouldn't necessarily just have to look at, one color of light, right? You'd image at several colors of light and then use our inbuilt spectral unmixing features to be able to say, yes, that is actually coming from my contrast agent and not, say, an increase in, I don't know, oxygenated hemoglobin or something like that. So those tools are available on the system for doing that. I would say, again, more specific, spectrally unmixed, where you've got, the optical characteristics of your contrast agent that you're using. You tell that to the system, and the system will find that contrast for you.
So this question
Yeah.
to me, it kind of feels like the difference between a pilot study and a study that you would publish. I'd say go ahead, inject your dye, see if you can see the difference between the uptake in the tumor and the surrounding tissue. Get confident that you can actually see what it is you think you're seeing. But then to Drew's commentary, being able to tell the difference of where exactly it's uptaking and talking about the validity of the science behind the probe. I would never say don't use a negative control.
I mean, you would use you'd have three. You'd have your labeled antibody, then you'd have your unlabeled antibody, and then you'd have your ICG alone, right? Those are your three control groups, or your experimental group, and then your two control groups.
Felipe, it looked like you wanted to add something there.
Now, yeah, you can also take a reference image prior the injection of the ICG and after the injection. You can do it in 2D and in 3D. And you can not only look at the ICG antibody, labeled ICG antibodies binding by running the spectral mixing, but you can also do some spectral analysis. So that's the benefit of this system. It does not only show images, but it shows as well the spectral properties of your specific compounds over the wavelength range. And that can be very precise to make sure and double check that what you are visualizing is really your ICG compound that is bound to the receptor.
Great, thank you. We have a question that's come in about contrast agents and the calibration sensor. So it says, is there a reliable contrast agent that users can routinely measure on the Vevo Laser X system to ensure that the laser output remains stable over time instead of using the calibration sensor?
If I can comment on this, there might be some confusion here. So the calibration sensor is there to measure the potential effect of the fiber or anything between the fiber and the animal on whatever you're trying to image. So that's what that calibration is for. If you're worried about variations in energy over time, we actually monitor the energy, every single pulse of the laser. We monitor that energy and record that value and correct for that that fluctuation. It's normal for, you know, pulse to pulse fluctuations within a certain range. And we actually apply a correction, a gain correction to correct for that. So you shouldn't really need to worry about, you know, energy over time. What I would say is if you want to sort of an external way to sort of calibrate or sort of understand the properties of your laser. There are some contrast agents. Actually, our applications group was just in Toronto not too long ago, and copper sulfate. Now, while you can't use this in vivo, you can use it in vitro. So we've got an in vitro imaging setup where you can introduce contrast agents and image those contrast agents side by side. And copper sulfate seems to be a pretty good one because it's very, very stable. It has very predictable absorption curve that doesn't change over time like some other contrast agents do. And you could use that just as it's more of a like quality control check than anything else, just to make sure that, you know, somehow the properties of your laser aren't changing over time, which they really shouldn't. And we do correct for those changes up to you know, to an extent. But if you want that sort of QC check, I would recommend copper sulfate. And please reach out to us because this was done fairly recently, and we'd be happy to provide some more information about, you know, the concentration or how to set up that control.
This question had several parts to it, but one area I wanted to highlight was sometimes they mention getting an intensity from the gel that's coupling, or that the signal from the tissue may be attenuated or potentially missing. Anything to control for or account for in terms of gel or maybe variances between the animals when doing this type of imaging?
You may get some variability between different animal models. So, for example, if you're using black 6 mice, there can be pigmentation in the skin, which can attenuate the light. So, photoacoustics, for those of you who aren't aware, is you're using pulsed nanosecond pulsed light to generate an ultrasound signal. So it's a hybrid optical ultrasound imaging modality, very powerful. You get the advantages of detecting optical signals, chromophores in this case, but you also have the advantage detecting with ultrasound that you can image those at depth with high resolution. But there are caveats, such as, you know, melanin will absorb, so that could reduce the amount of light getting deep into the tissue, for example. At certain wavelengths, water will absorb. Usually at those sort of longer wavelengths, water will absorb a little bit. That's where that calibration, you know, so-called external calibration can come into it, where you can actually calibrate through the ultrasound gel in order to correct for that that attenuation due to water. So that's one way you can say, okay, I'm actually correcting for that, so that now when I look at my data, I'm not looking at the influence of water on my my signal.
Great, thanks, Drew. So we mentioned earlier about you know, the high frequency transducers and the small tumor size that you can image, but how deep can we really detect an orthotopic tumor?
Depends on your animal models. You can go deep to 6, 7, 8 centimeters easily, or it can be very superficial. We have our higher frequency transducer at 71 megahertz, when you can really go deep to 60 micrometers if you wish. So the scale is very wide.
Kind of jumping back into the PA discussion, we talked about detecting hypoxia in tumors. Why do we think that's important?
Well, it's essential, I would say, because especially when testing a therapeutic treatment, chemotherapy as an example, you know that the chemotherapy can be efficient before the tumor hypoxia, but then during hypoxia, the tumor becomes more resistant to the treatment and also more aggressive. So you must qualify your treatment before hypoxia, during hypoxia, and before necrosis. Not all tumor turns to hypoxia, but majority of tumors. Cancers are very aggressive, such as the breast cancer and the pancreatic cancers, because the tumor turns very quickly to hypoxia. So this is essential, as well as detecting lipids and collagen into tumors. So this is now feasible using our brand new laser, the Vevo Laser X20, where we can detect lipids over 900 nanometers up to 1320 nanometers. So this is also very important when testing anti-cancer drug to really extract information about these parameters.
Is there a specific photo? Oh, sorry, Drew, do you want to add to that?
Yeah, I just want to add that, there's totally right, Philippe. I mean, hypoxia is crucial for, cancer, not just cancer biology, but cancer treatment. And hypoxia can really indicate, a lot of treatments actually involve increasing blood flow to previously hypoxic regions of tumors in order to actually get drugs there. So if you want to do, say, biodistribution studies, labeling drugs with some kind of optical contrast agents to be able to see how far they go into the tumor. But hypoxia is really a sort of key ingredient there. And as I mentioned, that previous study where it's not just hypoxia of a tumor, it's hypoxia of regions of a tumor. So you can have this, well oxygenated periphery in this hypoxic core of the tumor. And with our resolution, you can really pick out those subregions of even very, very small tumors in your small animal models. And the aspect too, just of the longitudinal aspect of the imaging, which Philippe alluded to with treatment, where you want to image the animal every day, and follow it throughout the course of treatment rather than having to, sacrifice individual groups and do histology. You can really follow with high resolution the progression of that hypoxic marker, in a living animal rather than a dissected, stained tissue. This is obviously has huge translational implications for developing treatments for ultimately for patients. And in a lot of cases, our customers are using patient-derived xenografts in their tumor models to mimic the human disease more closely in their animal models.
Giving me I was going to say we used to use it even just to figure out when to start treatment. So at treatments that are actually acting on hypoxic regions, you don't want to start those too early. You have to actually wait a certain amount of time, and each tumor is going to be different. Each animal model will be different. So being able to calculate that ahead of time, make sure that you're able to treat what it is you think you're treating, and really address hypoxia in total.
Yeah, it's really the sort of screening. That's a good point, Anna. The screening aspect as well. And maybe even go back even further if you're doing these sort of image-guided injections of your tumors or tumor cells in order to implant them. The system can be used as a screening tool to see when is the tumor actually starting to grow? Did the tumor did it take? Is it behaving the way, as Anna, you suggest, is it behaving the way we think it is? Is it hypoxic enough to start treating? So these are all really good screening approaches that can be used.
Great, thanks. Do we have a photoacoustic contrast agent that could be used for studying tumor growth over time?
Yes, there's one that has been tested successfully on different tumor type. It's the Angiostemp 800. It's targeted contrast agents targeting the alpha beta 3 integrins that are present during the formation of the vasculature. So you can use it as a tumor growth marker. It has been testing on brain glioblastoma tumors, on pancreatic tumors, liver tumors as well, and also bone metastases. So we have protocol in place also for setting the parameters on the system. And there's papers that have been published on the use of this contrast agent. But otherwise, you have a bunch of nanoparticles, such as these ones. These are gold nanostars that you can use as a functionalized contrast agent. And this is also perfectly good and even more photoacoustic compatible than any other organic contrast agent. And there's also now coming in the research new organic or inorganic contrast agents that are more sensitive in the far near infrared. So, which means that we now have good candidates that can absorb the light and emit a strong photoacoustic signal at 1,000 nanometers, 1,200,000 nanometers, and even higher. And this is perfectly good for us because we are in a wavelength range where we are not annoyed by the deoxyhemoglobin and the melanin content into the animal body. So we are in a process today testing new contrast agents, new candidates, absorbing light and emitting a strong photoacoustic signal over 900 nanometers up to 1320 nanometers.
Great. Thanks, Philippe. I think we have time for about one more question. We have one that came through the chat. Can you use photoacoustics on human subjects? How well do the results and the conclusions from photoacoustic imaging on animal models translate into human models?
Oops.
Yeah, it's an interesting question. So our systems are not approved for human use. They're preclinical systems for preclinical use. Um, however, there is a company out there who's doing photoacoustic imaging in the clinic. Interested to see some of the applications there. I believe that one of the targets of this company are breast tumors. So looking into breast tumors and measuring the oxygen saturation in breast tumors, and basically stratifying patient groups to see whether or not to perform a biopsy. So this is really to try and eliminate or reduce unnecessary biopsies for benign lesions. I would say as far as molecular imaging goes, there's work to be done. What I would say is that the analog is sort of optical imaging, where optical imaging using contrast agents has kind of made its way into the clinic in terms of surgical guidance. So labeling tumor tissue with some kind of optical agent, some kind of chromophore, and then using, say, fluorescence imaging during the surgery in order to remove the diseased tissue and leave the healthy tissue. I think that's one potential for photoacoustics where, rather than just being able to look at the surface during a surgery, you're able to see a little deeper into the tissue and be able to potentially identify tumor tissue that's been labeled with a chromophore deeper in the surgical field in order to resect that tumor. But as I say, our systems are not approved for clinical use.
Great. Thanks so much. Well, I think that's probably a great way to conclude our session today. I want to thank all of our experts for joining us today, and of course, thank all of you for joining today. Just want to remind you that we have a variety of resources available on our website. A recording of this webinar will eventually be posted, as well as all of our previous webinars. There's links to publications, to images. And of course, if you need any assistance throughout imaging, please feel free to reach out to us, and one of our extremely talented application scientists will reach out to you to answer any questions that you have.